So for today's class, we'll look at sample problems with respect to week 8's lectures. I hope you can see my screen. Yeah, so to start with, dash is the prokaryotic circular chromosomal DNA, and four options are given here: nucleoid, plasmid, nucleolus, and pilus. You can take a couple of minutes to think about this and then we'll discuss. Okay, so if we look at the structure of a cell, you have pilus at its extreme end. This is similar to your fimbriae, which are like small hair-like projections, but pilus is also used for transfer of DNA. Does anyone here know what kind of DNA is being transferred using pilus? You can try answering that if you know whatever you know about pilus. There are different methods of transfer of DNA from one cell to cell. You have first two classifications: vertical transfer and then horizontal transfer. Vertical as in from one generation to another, whereas horizontal is between two different cells. We have methods like conjugation and stuff like that. Is anyone aware of pilus? Okay, so this pilus is used to transfer F plasmid that confers stability and in some cases selection as well. So that doesn't that is not the DNA per se. Plasmid can refer to anything. and it need not be specifically prokaryotic. Nucleoid is that chromosomal circular DNA that we have in bacteria. Nucleolus is the region. So the right answer is nucleoid. Right, I've just given this as a text here. The next question is the bacterial dash is made up of peptidoglycan, and four different options are given here: plastids, cell wall, cell membrane, nucleus. You can think about this for a couple of minutes and then answer. Okay, from the name itself, we need to know that this has components of both peptide and glycogen, peptidoglycan. So we'll first eliminate the wrong options. Bacteria doesn't have a nucleus, so nucleus can be eliminated. And plastids are specifically present in plant cells, so that's also eliminated. Now it comes to cell wall and the cell membrane. So cell membrane is made up of a bilipid layer, not peptidoglycan. Whereas cell wall is the one that is made up of peptidoglycan, and this is what differentiates the amount of peptidoglycan differentiates whether it is gram positive or gram negative. So in the figure given here, we have the classification between two different types of bacteria. One is gram positive bacteria, the other is gram negative bacteria. Gram positive is the one that has more peptidoglycan, whereas gram negative is the one that has lesser peptidoglycan. Fine. So now the question is, which of these is made up of peptidoglycan? Which is your cell wall? Okay. Hmm. The next question is mesosomes. Mesosomes are also known as dash chloroplasts, chondroids, peroxisomes, and vacuoles. You can take a couple of minutes to think about this. Yes. So, do you want to try answering what mesosomes are? Do you know what mesosomes are? We'll leave the name, but
So their mesosomes, which is similar to the function to the mitochondria.
do they have any
So they are
specific function?
chondrioids. What's it?
Do they have any specific function?
Like respiration.
Oh, is it?
They
Okay.
play role in respiration.
Okay, from what I had read, it appeared like these are just invaginations, like when you bud off from within the cell, and this happens when you actually try to fix your cells, as in fix, as in freeze the metabolic state. At that time, from the membrane, because of this pressure, you have these small invaginations, and these do not have any particular function. These are rather called as artifacts. So, for example, when you want to image a particular cell, what you need to do is you cannot allow your cell to. Okay, there are two kinds of imaging. For example, when you add a particular drug, you can either allow it to grow it, continuously monitor it, but in most microbes that we are interested in, we cannot risk contamination. So what we do is we freeze them by adding a few chemicals. So they are metabolically frozen. So they can't grow. So we'll prevent that contamination. So when we do this, what happens is it might also stress out the cell because these are undergoing transformations, like chemically they are being modified in a particular way where they are not anymore viable, but their metabolic state is like frozen, like dormant. So what happens is, because of this stress or environmental pressure, you have multiple effects within the cell, one of which is this formation of these invaginations, like I have it here. So what they, sorry, yeah, so from within your cell you have these invaginations, like how you form these liposomes, or in any of the endocytosis process and so on. So these do not particularly have functions, but these are just artifacts of cell fixation. The answer that you told is right, chondrioids, but from what I read, I couldn't find any article or any information where it was said that it is involved in cellular respiration. You can maybe share the link here, so in case if I had missed it, I'll take a look at it.
Yes, sir. When I was preparing for GATE exam, I read like it is extension of the cell membrane also helps in a cellular response, but not particularly sure that all.
Okay. Is that an organelle then? Could it be termed as an organelle then? Does it like have any enzymes associated, or do you have any idea about that? Because I am not aware.
No idea, sir. I have only this much basic idea about the mesosomes.
Okay, okay. This is what I know. Fine. If you could share that article, we can have a look at it.
Okay, sir.
Sure. Yes. So okay. I've also listed down functions of other organelles. Chloroplasties are found only in plant cells and they are involved in photosynthesis. Peroxisome, it is found in eukaryotes. It will break down your fatty acids and is also involved in hydrogen peroxide detoxification with the enzyme of this catalase enzyme. And vacuoles, this is present in plant cells and some protists and fungi. It is used for storage and also gives turbo pressure in plants to ensure the cells are structurally uh, uh, the structural integrity is not compromised. Fine. The next question is, dash is not a eukaryotic cell. Bacteria, animal cells, yeast, fungi.
Bacteria.
Yeah, this is pretty straightforward. Yeah, the major difference is the presence of a nucleus. You also have some more differences, like, for example, the presence of multiple other organelles and embedding of ribosomes to your Golgi network. Whereas here it's all just distributed and the organelles are not very well defined, like it is there in eukaryotes. And next is a question that we just a practical application. Microbiology media are autoclaved at dash for 20 minutes. 100 degrees Celsius, 121 degrees Celsius, 10 degrees Celsius, and 58 degrees Celsius.
121.
Yeah, what is this process called as?
Autoclave.
Yeah, so this process of sterilization is called
Sterilization.
autoclaving. Yeah, and do you also happen to know at what pressure we do this?
I don't know, sir. And one, I think so.
So
Or five.
units.
PSI.
Yeah, so it's actually 15 psi.
Okay, okay.
Yeah, so it's 15 psi, 20 minutes, 121 degrees Celsius. That is the autoclave cycle. Dash is a commonly used media for the maintenance and propagation of Pichia pastoris and Saccharomyces cerevisiae. And we have four options: YPD broth, CSM media, YP gal, YNB. So here again, we can take a couple of minutes to think about this. We'll start by eliminating the wrong options here. So if you know of any incorrect options, you can start with that and justify why that option will be incorrect. That way, we'll go through all the uh, options. first. So CSM media is a complete supplement mixture. So here they use it with engineered yeast strains that require specific nutrients. For example, if we want to study a particular metabolic pathway, we usually do that by disrupting that pathway. For example, knocking out a gene. In those cases, if it is if that gene codes for an enzyme, then it will not be able to produce a particular metabolite. In those cases, we need that metabolite for its growth. In So typically the CSM media is used in that cases. You might have heard of oxotrophs of methionine and so on, like for essential amino acids. So this complete supplement mixture will have a percentage of all the essential metabolites in such a way that this can be used to select or grow the oxotrophs. And YP-gal is yeast extract peptone galactose. Here it is used, galactose is the source of sugar. So this again depends on what you want to do within the cell. Like for example, in yeast you have galactose based promoters, which means that galactose will induce your protein of induce the expression of your protein of interest. So in cases where uh, the plasmids that have these gal promoters, you can't use YP gal. The next is YNB, yeast nitrogen base. This again, it's a minimal media. Again, it's used for oxotrophic selection, like we mentioned in CSM. The only difference is in CSM you'll have for all. In YNB, you take yeast nitrogen base that gives you the minimal sources of carbon, nitrogen, sulfur, oxygen, and phosphorus. And other than that, you need to add the supplement. Like for example, in methionine oxotroph, you need to add methionine separately. That is what is given as requires supplementation with amino acids and other nutrients. Now coming to the right answer, which is YPD broth. This is commonly used for maintenance and propagation of Pichia pastoris and Saccharomyces cerevisiae. So this is yeast extract, peptone and dextrose. Now here your dextrose is the source of carbon, whereas peptone is your source for nitrogen. And because you have peptone and dextrose, this is a rich media, although the composition is not very well defined because you have this extract, but it is based on observations and based on growth, it is set to set to propagate multiple different strains. Yeah, so YPD is what we use for maintenance and propagation. And the next one is DASH is a mammalian cell culture media. And we have four options: DMEM, tryptone broth, YNB, Hinks TNMFH media. You can take a couple of minutes to think about this and then we'll come back to this. Yes, do you know what DMEM is? The full form and the components by any chance?
Delbec's modified Eagles medium.
Yes, good. Huh.
It consists of fetal bovine serum and amino acids, and I don't remember much of it.
Okay, okay. Uh, what else do you know about this? What is the kind of cells
It
that
is
can
used
grow
for
on this? Yes, sorry, you continue.
mammalian cells.
Huh. Okay. And any idea about the carbon source here? Which carbon
Glucose,
source? Yes.
energy source is glucose.
Yes, yes. And is it a complete media? Complete as in, is it like what we saw earlier? CSM. We saw something here, right? CSM. Is it like CSM? Is it like CSM in terms
I
of the
don't know.
composition? Okay. Based on what you answered, you can try to think, because you told that it has, okay, it doesn't really have fetal bovine serum. When we grow mammalian cells, we add fetal bovine serum in addition to the DMEM media. It is not present in DMEM.
Okay.
So, what is this fetal bovine serum? Do you have any idea? Is it a protein, or what could be the what role could fetal bovine serum be play?
Uh, it provides growth factors, hormones.
Okay. Yes. So, like you mentioned, it provides growth factors and hormones, which are essential for mammalian cell growth. DMEM doesn't do that, so it is not a complete media. Good. Do you have any idea about other broths? We saw about yeast nitrogen base. One more thing is that. Okay, we'll first go through the answer. It contains glucose, amino acids, vitamin salts, and others. Yeah, did you want to say something?
No, sir.
Okay, okay, no issues. Okay. While tryptone broth is what we use for bacterial culture, uh, that alone cannot supplement the growth of mammalian cells, which is why we can't use that. And this Hinks TNMFH media, this is for insect cells or plant cells, not for cell culture. Fine. Okay, we come back to the related question. What is the role of FBS in cell culture media? And like you had mentioned, it provides growth factors and hormones. So the options that are given are to supply nutritional content, to help the cells adhere to culture dish, to help the cells grow in layers, to help the growth of organoids. So we have, although you had mentioned that it helps in uh, providing hormones, and but do you know?
And it also helps in providing attachment proteins, so it gets adhered to the culture dish. I remember it.
So FBS provides you proteins that helps your mammalian cell to adhere to the dishes. That's what you're saying, right?
Yes, sir.
Okay. So do you know the different kind of cell types that we have? One is adherent. The
And
other,
free, like suspending cells.
yes. So in
Uh,
those cases, will we need FPS?
for suspending cells, sir.
Yes.
I think no need. I think I have 50-50. I think, sir. Like
Yeah, it could be
attached.
one of the both. Huh.
Yes, sir.
Huh. So I am not sure. I have not come across this thing where it provides proteins for adherence. These proteins are naturally synthesized within the cell, and your dishes have a complementary interaction providing interface. So unless your cells naturally have these adhering surface or proteins acquired for adherence, it can't bind to the culture dish and grow. So FBS does not provide that. It just provides you nutrients. And so option B we can is actually incorrect. And do you know what organoids are?
They are miniature organs. It mimics the original organs only, but
R-
it is in miniature form.
Okay. Yeah, these are again tissues, very small tissues, but like they are supposed to mimic organs and usually used in developmental studies. Um, yeah, right. Now, dash antibiotics are commonly used in mammalian cell culture media. Ampicillin and kanamycin, penicillin and streptomycin, penicillin. So, uh, you can think about this again, and you can also think why a particular antibiotic or a combination of two antibiotics can be used.
Sir, penicillin is for sure, sir. I don't know whether it is with combination streptomycin or not.
Oh, why would you think we need penicillin?
So penicillin inhibits the cell wall synthesis of the bacteria.
Okay. Do you also happen to know about streptomycin? So that way we can get
Like
rid of
I remember,
bacteria.
like it doesn't synthesis 30s ribosome something. I don't remember exactly.
Okay. So, um, why not ampicillin, kanamycin?
Ampicillin is used for cloning purpose, selective cloning. I mean, selective, what do you call it?
Selection of bacteria that take up a particular clone, of course. What if we have no? Does it mean that we cannot use ampicillin?
Mm. Sir, for prevention of contamination, we use penicillin, but ampicillin we use for selective breeding, sir.
Yeah, yeah, that is true. That is true. Ampicillin, kanamycin, chloramphenicol. We have multiple zeosin and all these antibiotics. That one reason is because you have those resistance genes in particular plasmids, and we use that. But is there any specific reason why penicillin and streptomycin are only used in mammalian cell culture?
No idea, sir.
Okay, so like you mentioned, penicillin targets the bacterial cell wall, and it's again it is targeting gram-positive bacteria. Streptomycin, like you mentioned, it inhibits protein synthesis and is effective against gram-negative. This way, any kind of bacterial contamination can be prevented in mammalian cell culture, because usually uh, like let's say you uh, because the media is rich here, the and your mammalian cells take more time to grow, your bacteria can outnumber your mammalian cells in a shorter time because your bacteria have a shorter half doubling time. So you definitely do not want contamination from bacteria. That is why you want to add these antibiotics. That is clear. And penicillin for gram positive, streptomycin for gram negative. Was that clear?
Yes, sir.
Okay, good. Like you mentioned, we know that ampicillin and kanamycin, these are used in cloning experiments. Good. The next question is, filtration of cell culture media is done to dash the media. And four options are given here: sterilize, froth, solubilize, and anticoagulate. You can think about this for a couple of minutes. Okay, so filtration here is a process where we try to remove um, particulate matter because we make the media. Okay, there are two ways of making the media. You either buy the liquid media or take the powder and um, you add all the components, but you cannot really autoclave here. The question that one might typically ask is to sterilize components, you can autoclave them, but when you autoclave them, most of the proteins or essential nutrients would denature, and to prevent denaturation, we instead of autoclaving, we would just sterilize and store it at lower temperature. So this filtration is of cell culture media, it is done to sterilize. Frothing, solubilizing, and anticoagulating, none of this can be overcome by filtration. The next is for Drosophila in vitro cell culture, what kind of media is used? Of course, we can't use Delbeco media, it's actually DMEM, whatever you had mentioned earlier. So, this DMEM media can be used for mammalian culture, peptone for bacteria, insect for uh, insect media. So, Drosophila media is the one that is needed for Drosophila cell culture. Hmm. Dash does not contain DNA. Option A nucleus, Option B, mitochondria. Option C, chloroplast. Option D, peroxisome.
Option D, sir.
Option B or D?
Peroxisome.
Peroxisome, right. So like you mentioned, nucleus is are contains the core chromosomal DNA. Mitochondria has its own DNA, although it is thought of as uh, being acquired from an ancient ancestor organism. Again, chloroplast has its own DNA. Do you have any idea what the mitochondrial DNA and chloroplast DNA encode for?
What, sir?
Do you have it?
Mitochondria.
Yeah, so you're saying that mitochondria and chloroplast also have DNA. What does the DNA contain? In the DNA in mitochondria and chloroplast, does it code for any proteins or enzymes? Any idea?
No, sir.
Okay. So all of these are different organelles and they'll have need specific enzymes to function. So for example, in chloroplast, you have chlorophyll and photosynthesis happening. You need enzymes for that. So the enzymes that are encoded for chlorophyll formation and the processes involved in inside the chloroplast, those enzymes are encoded by the DNA within the chloroplast. Similarly, in mitochondria as well. And like you mentioned, peroxisome doesn't have its own DNA. The next is ribosomes are pelleted down at dash the 300G, 30,000G, sorry, 300G, 3000G, 30,000G, and 300,000G. So, to start with, you can mention what ribosomes are, and then um, if you know the right answer, you can also tell why that could be the right case. We'll start with what ribosomes are. You can think an answer.
Ribosomes are useful for protein synthesis purpose.
Yeah, are these organelles?
No, sir.
They are not organelles.
Because it is present
Ribosome
in
is not an organelle.
ribosome.
You can think, no
I
issues.
think,
You
yeah,
can think.
okay.
Yeah, yeah. Okay. So, do you have you ever used a centrifuge?
Yes, sir.
Okay. What did you use it for?
For separating the contaminants and the uh, like I use for cells like supernet and pellets, sir. So separating the bacterial cells from the contaminant proteins.
As in, like in protein purification, is it?
Uh, to collect the cells from the what we call it?
So you have a secondary culture, a primary culture, and overnight culture. To pellet down the cells, you had used centrifuge.
Yes, sir.
Okay. Do you remember what RPM you used it at?
If, when I was doing like 7,000 RPM, like 7,000 I used.
Okay, 1000 RPM. Okay. So with that, you could remove the cells. Now, why do you have any reasoning or any rationale behind why you use 7000? Can you use something like 700?
No idea. I think it doesn't separate.
Yeah.
At certain point, it gets separate when we use a certain RPM. It
Hmm.
gets separate like pellet and super.
Yes. So what decides this? What?
What, sir? What?
So what factor decides the RPM that you use? You told me that only at certain RPM it gets separated. Does it have to do with?
Centrifugal force.
Yes, and Centrifugal force is the result of the RPM, right? But
Yes,
is there something within your sample that decides this?
no idea.
Okay, it's the mass or the size of the cell. The larger your cells are, the lower the RPM you need, and vice versa. So, if you want to isolate very small components, you need to use very high RPM. So, what people use is something known as differential centrifugation. So, they break down cells, and different organelles have different sizes, and proteins also have different molecular masses. So, let's say you have different sized molecules here. In your first, you will use a very low RPM where higher molecular weight, like for example, cells are pelleted as debris. Then you increase the RPM. So gradually you increase the RPM. And based on what was observed, because ribosomes are organelles, not as large as cells, they need an RPM that is more than uh, 7,000 for sure. So option A and B we can remove. Now, is it C or D? That's a question. Now,
Sir,
with
RPM and G is different, sir. Like RPM and G
yeah, yeah, yeah.
units.
You just need to multiply it by 9.8, right? RPM and RCF.
Okay.
Is it not that case?
No, I always use it in RPM, so that's why I didn't remember G. What's G?
Okay, okay. So it's just that you RP. Okay, there is one thing called as RPM, one thing called as RCF. So RCF takes into account of your this force due to gravity. So you just need to multiply it into 9.8. So I think in all the centrifuges that people use, they have this conversion. Okay, we can even consider this G as RPM because you are familiar with that, although it's slightly different. Yeah, so now let's say 300 and 3000 are removed. Now, will it require 30,000 G or 300,000 G? That is a question. So, typically for organelles, up to 50,000 G are used. And in this option, because 30,000 G is given, we'll go with 30,000 G. Huh. Was that clear?
Yes, sir.
Okay. The next question is periplasmic fraction is isolated to dash. Option A purify periplasmic protein. Option B to isolate cell organelles. Option C to isolate the nucleus. Option D to isolate mitochondria. Yeah, so to start with, we can decide discuss what periplasm is, and then if there are any, what is the content of a periplasm. With that, we'll be able to answer this question.
So periplasm means it is space between the inner membrane and outer membrane of the bacteria.
Right. So, um what will be present within this inner and outer membrane? What kind of molecules will be present?
I mean between these both membranes, sir. I mean
Yes.
say,
Yeah, yeah.
like enzymes.
Yes. So, proteins. What else could be there?
I remember only enzymes. I don't know what else are there in that.
Okay. Um, right. So, will it be helpful for isolating cell organelles or nucleus or mitochondria?
No ideas.
Okay. So, the thing is, um, your cell organelles, which includes your nucleus and your mitochondria, are just held inside your inner membrane. It is not between your inner membrane and your outer membrane. You need your periplasmic fraction if you want to identify something that is stuck to your inner membrane and outer membrane. So this is how it looks like. So mostly for secreted enzymes and chaperones, and we take the periplasmic fraction because these have a tag that allow it to go through the periplasm to the outer membrane. And some, in many cases, proteins that are expressed on the surface of the cell are usually folded in this periplasm because it has this reducing environment that allows it to be folded. Now these are what are usually majorly present in the periplasm. So for to purify this, we use the periplasmic extract. Yeah, and it is present in gram-negative bacteria. Yeah, so I hope you understood what are the kinds of enzymes that we'll have, or what are the kinds of proteins we'll have here.
Yes, sir.
Yes, and we come to the last question for today's class. In cesium chloride density gradient centrifugation, the order of gradient components is dash, and four different orders are given for DNA, RNA, proteins, and organelles. So, to be able to understand this, you need to know what cesium chloride density gradient centrifugation is. So, do you have any idea about cesium chloride density gradient centrifugation?
No, sir.
Okay, you know about normal centrifugation, right?
Yes, sir.
Okay, so in your normal centrifugation, uh, your cells, like for example, in the case that you did, your cells are more dense than your media. What media did you use? LB or something like that? Luria broth, maybe?
Yes, sir.
Yeah,
It'll be pro.
yes. That is less dense than your cells. That is why your cells will pellet when you do the centrifugation. Right. Now, uh, let's say we want to isolate specific compounds with specific density. So in those cases what we do is we add different density layers in such a way that when we spin, compounds will segregate and stand in their particular in a density that is equal corresponding to their density. This is called as isopicnic point. You can maybe take a look at it later. So let's say you have a combination of, when you lyse the cell, DNA, proteins, organelles, and RNA, um, at extremely higher RPM, all of this will settle down. But if you want to specifically isolate fractions, what you can do is you can take this cesium chloride, add different densities of cesium chloride in such a way that it can form these this gradient. And when you spin it, depending on the density of your particle of interest, it would be associated with a particular layer. You extracted DNA or RNA earlier?
Yes, sir. DNA I have extracted, sir, from
Yeah,
the cells.
yeah. So have you used this phenol chloroform isoamyl mixture?
Yeah, to precipitate the DNA, we use it, sir.
Yes. So even there, you might have seen two layers, and only one layer will have your macromolecule of interest. Even that is a case. I mean, although that does not actually use this density-based segregation, but rather you want to preferentially separate your proteins from your nucleic acids. In a method similar to that, having with two densities, I mean, having different solvents with different densities, you will be able to isolate specific molecules of interest. So that is what is shown here. Um, in this particular case, when we have in cesium chloride density gradient centrifugation, the heaviest one will go to the smallest region or the shortest region. So it will be organelles followed by proteins followed by RNA and DNA. That is what is given here.
Sir, can we separate at a time like all the DNA, RNA, protein organelles at a time by
You,
using C?
yeah, you can. That is what. So have you studied about this I think it's called the Michelson-Stahl experiment, where ammonium chloride and its isotope was used to be able to identify how this process of semi-conservative replication happened. Do you remember that
No.
experiment where there was this group of scientists called Michelson and Stahl, a couple. They were a couple of scientists. They were using, so they wanted to identify if the two new strands of DNA are from the original DNA and how this replication happened. So for this, they used an isotope of ammonium chloride as the nitrogen source, because you have nitrogen being incorporated within the DNA. So there also they use this cesium chloride density gradient. And to answer your question, it is possible to be able to do this. It's just that you need to have a rough estimate of what the maximum density and the minimum density would be, so that you will have gradients. Because first you need to make this gradient, and making this gradient, I just recently read about it, how to make these gradients. So what I could understand is you need to make solutions with different densities, and once you achieve the solutions with different densities, independent of whether you spin it or mix it, at the end they will all stand according to, or be arranged in layers according to their density. It is similar to adding oil to water, or water, petrol, honey. You might have done these small experiments when you were in school, where even after mixing, they would all be immiscible and separate according to their density. Uh, do you have any idea about these experiments? Like small
Yes,
schools.
they form emulsion.
Yeah, yeah. In some cases they do mix, but in most of the cases they segregate and form these layers. So depending on the density of your macromolecule or your organelle, it will be present in one of the layers. It's just that you might not be able to visibly see them, because unlike pelleting, where they're all getting to the bottom of the um, your falcon tubes, here they might be suspended. It might be precipitated. We don't really know, but you will be able to definitely isolate molecules, macromolecules that have the same density. That is for sure. As long as your these components have different densities, which is true, you'll be able to separate them. Does that answer your
Yes, sir. Okay, sir.
okay? Yeah, okay. So with this, we come to an end for today's class. If you have any specific questions, you can ask me. I'll stay on the call.
A lot.
Yeah, yeah. So what does ribosome do?
Protein synthesis.
Yeah, what are
Why I said no,
organelles?
it is
Yeah, sure.
because in a prokaryote cells, it doesn't contain organelles, no sir, but it has ribosomes.
Yeah, it is one of the fewer organelles that your
So in
prokaryotes
that
contain.
way, I thought in that way, it said for eukaryote also, I think it's not an organelle. That's the reason.
Okay, okay.
It is also non-membranous. Double membrane doesn't have double membrane.
Yes, but it is present on. Okay. Ribosome is a mixture of proteins and RNA. There are these RNA called ribosomal RNA. These are rRNA. You might have studied it. This ribosome, it's actually it is a very big complex that has multiple different proteins and different RNA. So although, because you mentioned that it doesn't have membrane, which is true, it is considered an sort of organelle because it does the job of protein synthesis and it is intact. Does that answer?
Yes, sir.
Okay, okay.
That's it. Thank you.
Thank you.